So hopefully you had a good immersion to metabolomics yesterday and probably raised some questions to you. Since my talk started very early in the day before you had a chance to think about workflows and approaches, this is an opportunity to revisit the topics again and in a sense to recall and reinforce what you guys need to do right before submitting samples to the core. So there's going to be a lot of repetition, excuse me on that, but this is important repetition so that you guys understand what needs to be done, and we'll talk about the various workflows and approaches today, okay? So this talk is going to deal with the different model systems, again, the cell, animal, and human studies, how to design a good study for them, and how to effectively collect samples, store them, and process them. So we talked about cell systems again yesterday. We know the advantages of a cell system being its quick turnover, it's a homogeneous population. You can do an in-depth study of the cell metabolites and its responses to your interventions. It's very easily replicated. You can do with small sample size in this situation because of the very homogeneous nature of the cell population. And since you control everything in the media, the nutrients you give, the ambient atmosphere and everything, the biological variation is much less, therefore you can have small sample sizes. And the technical replication of these samples is also very good. It's also very ideal for flux studies because you're studying one cell type and you're able to put in a certain labeled metabolite and follow it across the system and therefore gain more knowledge about your biological question without much interference from other issues. So yesterday I briefly mentioned about endometabolome and exometabolome. Basically, studying the endometabolome is just studying the metabolic fingerprint, what's happening inside the cells, how your metabolites are being handled. Studying the exometabolome, on the other hand, is studying the secretome of the cells. What are the cells secreting, how it is changing the media around it, essentially the metabolic footprint of the cells. To process these samples, especially the exometabolome, you aspirate the media after your intervention. You try to remove the cell debris by centrifugation and passing through filters. And we typically try to concentrate these media by either drying with vacufuges or nitrogen drying. And we use various cases of solid phase extraction as well to concentrate and focus on the metabolite you are interested in the metabolome. The endometabolome can be treated in many ways. Yesterday I shared with you a way that we use in our core, which has been validated by our studies, but that's not the only way, of course. We can, a cell type can be quickly washed and either directly extracted, quenched, and detached in one step using solvent system, or you can decide to quench with liquid nitrogen and detach and then extract, as I mentioned yesterday. Other option is to detach first, give a quick quenching with the appropriate solvents and use the washed pellet for analysis. So there are different ways to approach the problem. There's not just one way. And sometimes depending on your cell type, we might have to deal and make compromises to pick the different kind. Mm-hmm. The problem is usually the signal strength, and if you're looking for low-level metabolites, probably it's okay. But we probably have to do some pilot studies to exactly see whether you're getting the answer you want. But more the volume, the better, of course. You can always pool things if that works for you. Yes, I understand. We can work with you in concentrating sample sizes. Like once we load in the mass spec, we can control the concentration of the sample that's loaded in the mass spec. Typically, we use 50 microliter to 100 microliter. But if sample size is an issue, we can cut it down to 10 microliter, 20 microliter, therefore concentrating your sample. So we can work with those things. But ideal would be 10 million. That's perfect. We don't need to do much modifications to a protocol. But if your sample is precious and it's small in size, we can concentrate things down to do things in the mass spec. We just won't get many technical replicates of the same sample. Mm-hmm. I don't think specific literature is out there for collagenase, but trypsin is generally frowned upon because it kind of alters the metabolome a little bit. But again, if you're doing it throughout your experiment, you're eliminating that bias. So if you're using collagenase throughout the experiment, again, you'll be eliminating that bias. Okay. So this, I want to show you the study we did from a core-affiliated lab. This is insulin producing beta cells that we studied TCA cycle and the glycolysis metabolites. This is the effect of using a water rinse versus a Krebs ringer buffer rinse. And as you can see, the water rinse shows much more capture of the metabolic profile than the KRB rinse, which is specific to this cell line. So water rinse is better in this situation, but how long of a water rinse? Even that makes a difference. A quick 5-second water rinse, which we call rapid, or a 30-second water rinse also shows some changes in the metabolic profile. I don't want you to get scared about the small variations causing changes, but the point here is to standardize your protocol. Whatever you choose to standardize throughout your experiment. So what about quenching? In this experiment, Mathew compares liquid nitrogen minus 80 and 0 degree solvents. And as you can see, the white bars are the liquid nitrogen, the gray ones are the minus 80, and the dark ones are the 0 degree. And you can see in certain metabolites, this quenching procedure causes differences. So in our core, we recommend liquid nitrogen quenching. It's quick, it's achieved very easily. But if your cell requires a modification to the protocol, we will work with you. This is different extraction solvents. One standardizing the rinse and then the quenching. What about the different extraction solvents? How much coverage of the metabolome are you getting? So these are again the glycolytic and TCA metabolites. And you can see the variation in the pattern based on the organic solvent you choose. This is methanol chloroform combination, 9:1. This is pure methanol. This is methanol triamine chloroform combination. So depending on what your question is and what the extraction solvent will answer your question, we might have to modify your protocol as well. So we discussed yesterday what are the things that cause variability in a cell culture. Complete control and dependence of nutrients is a characteristic of a cell culture. So you control everything about the cell system, and whether you capture it at growth phase, at stationary phase, if you forget to change your media and stretch it from 48 to 72 hours, there will be effects to your metabolic profile. So standardize the time and adequately replenish the nutrients that you are wishing to give to the cells. This is especially true of primary cells. They're very delicate. Growth cycle, you time your sampling according to the growth cycle you're interested in and be consistent about it. Medium composition, generally most people use the same media throughout the experiment. That's not the issue, but batch effects do matter. If you buy media at a certain time and then you go back and order from the same vendor, sometimes the consistency does not exist, so stick to the same batch of media. We talked yesterday about centrifugation speeds as well. The point is to capture the endometabolome very well. Therefore, low centrifugation speeds are essential so they don't lyse your cells, therefore contaminating the exometabolome and not capturing a good endometabolome as well. So we'll talk a little bit about animal studies. As animal studies essentially work, they are existing in your lab control environment. There is a reasonable control of what you do to the animal. The advantages being that you have a whole organisms to work out, kind of replicating human situation. You can do time series studies, interventional studies, do adequate control studies, and there's options to get various tissues and biofluids from the animals. We mostly work with rodent studies, so I'm focusing on that, but of course we can work with other animals as well. Blood collection usually is very hard to do with small animals, but you can use microwave tubes like this. These are EDTA plasma collectible tubes. You can collect the blood and centrifuge them in the same tube and store them in -80 after taking the supernatant. We can either use a tail vein bleed or a tail bleed or a retroorbital collection of blood. The volume here is quite low. We understand that. And certain assays, it's okay to have low volumes. But if you're aiming for larger spread of recovery and you are running multiple pooling of samples, it's not a bad idea in this situation to get the volume you need. Urine collections, we talked about the metabolic cage yesterday to separate the feces from the urine. Also, if you're simultaneously collecting tissue, you can also do a direct blood aspiration. If you're going to do routine urine collection throughout your study, you can do collection on plates or on plastic wraps so that you separate the fecal material and use a pipette to gather the urine. Please collect in sodium azide containers, centrifuge, remove the debris, and store in -80 again. As I said earlier, tissue collection from animal studies and multifold options, you get various organs that you can sample. It's important to practically gather supplies when you do this because you want to achieve the whole opening from euthanasia to collection to snap freezing in a very restricted time frame and try to maintain the time frame consistently across your study. Be sure to sterilize your field so that you have the same setup every single time. Collecting tissue, either the whole organ or needle biopsy can be achieved in this. We talked a little bit about residual biofluids. If that's an issue with you, you can either try the whole body perfusion, we explained through the left ventricle, or you can decide to wash your piece of tissue and saline to remove the interfering biofluids. Snap freezing in liquid nitrogen should be immediate, so collect a liquid nitrogen container next to your collection. And we should achieve everything in a reasonable time frame, in a reproducible time frame. So again, variation to tissue collection are fasting time. We usually decide to stagger even the removal of the food containers as you're working with different animals. Sleep light cycles in animal rooms and microbiome changes are different in different rooms in your animal area. Therefore, stick to animals in the same area. That matters too. Metabolic cages and containers can sometimes be cleaned with disinfectants. That might be the huge peak in your analysis. So make sure to take notice of what's going on with the biofluids that touch different parts of your animal cages. And we'll talk a little bit more about mode of euthanasia. Yesterday, I told you guys that the mode of euthanasia makes a big difference. This is a study done by Charles. Here he studies skeletal muscle, heart, liver, white adipose tissue, and serum with different modes of euthanasia. The red ones are the one with the cervical dislocation, so a hypoxic form of euthanasia. The green ones are carbon dioxide, and the others are the varied other anesthesia agents. As you can see, consistently the hypoxic form separates out. So does the carbon dioxide euthanasia. So the mode of anesthesia does matter. So do not change the mode of euthanasia in the middle of an experiment. That would be a big bias. If you're sticking to one, depends on the question at hand. Hypoxic patterns do tend to shift the metabolic profile to hypoxic patterns. So tissue collection and anesthesia with the circulating blood seems to deviate away from the effects of hypoxia. If you choose carbon dioxide or cervical dislocation as your euthanasia mode, this would be a factor to consider. Human studies, of course, we get the closest information about the disease at hand, the question at hand. Often interfering are the genetic and the environmental variability of human studies. It's important to really collect sufficient metadata from these patients. So the time of diet, what kind of diet, what time did you get the sample, who collected the sample, all those things matter with human studies. especially diet and timing of the samples really, really matter. This is evidence to show why serum or plasma might be important to your studies. This study compared serum and plasma from the same individuals across the board and splitting them into different varied groups of metabolites. As you can see, most of the metabolites do correlate with each other with the serum and plasma, but phosphatidylcholines, some amino acids seem to have issues with correlation with the serum and plasma. And if your metabolite of interest is not within these categories, it's perfectly okay to use either serum or plasma. But if you're doing a whole targeted profile to see what's different, you can see you will be losing out on these patterns when you use serum versus plasma. So plasma collection, if you are planning a study which does include most of the metabolite classes, use EDTA plasma. We talked about limiting hemolysis yesterday. Limit processing time, this is very important, from collection to placing on ice to going to -80. And also aliquot in small quantities, therefore you're limiting freeze-thaw cycles. And if possible, if you're planning a metabolomic study, already aliquot a separate one for us. So there is no need for further freeze-thaw cycles. This is a slide about urine stability. In this study, they were creating a biorepository, so they sampled 40 healthy volunteers. Here I'm showing 4 different patients, numbered accordingly. And they compared storing urine immediately in -80 with storing for 24 hours at 4 degrees centigrade before getting to a -80 freezer. The red triangles are immediately stored in -80, and the blue ones are the ones stored after a day into -80. You can see already there is some changes in the time, in this small time frame to the metabolite profiles, causing separation between samples, which are technical replicates of these patients. So the time and the speed to get to -80 might need to be standardized and be as quickly as possible. We talked extensively about urine collection yesterday. Ideal would be 24-hour collections, taking a sample from a mixed 24-hour collection, which is kept on ice. It's important to instruct the patients on the exact mode of collection of samples. The next best option would be a first morning because it's as much far away from the last meal. Also, random midstream might work depending on the question asked. Definitely, we need to centrifuge these samples to remove the cell debris and other artifacts that you get in urine collections. Aliquot immediately on ice, store in -80, and transport on dry ice. I keep repeating store in -80 and transport on dry ice. That's because it's really, really important. If you send one batch with not enough dry ice to us and the next batch with adequate dry ice, you're already creating a bias in your samples. So this is important, hence it's repeated a million times. Okay, talking a little bit of sample size, as I mentioned yesterday, the more variables you measure, the more sample size you need. That's because we want to reduce the false positive rate and keep a good stringent false discovery rate as a cutoff. If your samples are from cell or animal, the biological variability might be less, and therefore you can do with small sample sizes. But usually human studies, the variability is so high that you need larger sample sizes. The one effective method we mentioned yesterday was to do a small pilot study to see if your effect size is huge enough to do with smaller sample sizes, or you would need a large sample size to show the effect size in your biological question. Okay. So we'll talk about the different study design approaches. We spoke about case control studies, we spoke about interventional studies, we spoke about crossover studies. Those are things you normally do, so I'm not going to dwell too much on it. But how do you use those studies with the metabolic approaches that we have? That's the question at hand. We briefly mentioned throughout many talks yesterday about targeted and untargeted approach. So depending on a biological question, you might start on top or the bottom, or do a combination of both. So if you have a biological question and the question is very driven by just the disease process in hand or the intervention in hand, you would start with an untargeted approach. You find the markers of interest and then focus on with targeted analysis. Sometimes you already know the hypothesis that you want to prove. Then you would start with a targeted analysis And to find correlating factors, go back to untargeted analysis as well. So this is an interchangeable workflow. We talked about Fluxomics yesterday. I'm not going to talk too much about it today because we're dedicating an entire half day tomorrow to talk about this. But this is a very good way to measure where your metabolites are ending up and what's causing the snapshot that you're getting, that stationary phase versus the flow pattern in the metabolite. Cell studies are really good for studying individual cell metabolite patterns. We can do it in animals and humans. It just gets complicated as the organism gets bigger and needs a very good consultation and planning to do flux studies. So definitely, I recommend that you sit down and plan a flux study very well with us to understand what the issues are before proceeding to plan flux studies. And as Charles mentioned yesterday, you definitely want an idea of where the metabolite snapshot is before planning flux studies. This should not be your first step into metabolomics, because this gives you additional information, but in a complicated, the data that comes back at you is very complicated and very diverse. So what about the other omics studies? We are talking about the metabolome, there's a proteome, there's a transcriptome, the genome. All these layers will add strength to your biological question or the answer you find. So how do we go about it and integrate it with metabolomics? So we have the three approaches here, the targeted, the untargeted, and fluxomics. As I told you earlier, these are interchangeable in direction and layers of support being added. Also, once you have some markers or answers from metabolomics experiment, you can always add layers of evidence and confidence by alternate biological methods like Western blotting or Luminex measures or ELISA measures. Other option is to use large data sets that you generate from other omics platforms, genomics, transcriptomics, proteomics, and you vertically integrate with our untargeted platform and you generate more evidence and more confidence to your biological question. Other option is to use other model system. You started off with a cell system, you proved it in an animal system, then you move it to a human system, or the other way around. You started with the human system, you move to an animal system, and in varied ways. I'm going to show you some examples of how this vertical integration of omics is possible by showing some examples of some core-affiliated studies that have been using these techniques. So this is a study from our group here. This is a study about progression to ESRD or dialysis in 40 patients with diabetes and 40 patients who progressed with diabetes to dialysis. So it was a nested case control study. We started off with an untargeted approach. We used an untargeted platform to quantify all the metabolites. And we did a volcano plot to see which metabolite causing the most fold change with the most highest p-value. And we found this marker called pseudouridine, which is a pyrimidine metabolite. We chased this metabolite further by quantifying it with a targeted platform. We didn't have a methodology for this. We developed a methodology, measured this, found the factor to be true even in targeted metabolic analysis. We wanted to know, being nephrologists, where the pseudouridine is coming from, what's happening to it. So we designed an in vivo metabolic study that is in animals. We injected a labeled precursor to pseudouridine, which is uracil, and we tagged pseudouridine in the plasma. We tracked the label incorporation across time. We did that in tissues, which I'm not showing here, and in urine to conclude how the flux of pseudouridine in a diabetic animal is. So this is completely possible depending on the question you have, whether to shift from untargeted modes to targeted mode to flux, or vice versa. How about the microbiome? This is also another core-affiliated study where the question was what is causing the C. diff susceptibility in animals after an antibiotic exposure. So when comparing the microbiome to the metabolome with an undirected clustering, you can see how the different phyla of bacteria and clustering of metabolites across the different KEGG pathway taxonomies. So you can see how these bacteria are able to change the microbiome and also change the metabolome, explaining the susceptibility to C. diff infection. So this is another example of combining omics studies, microbiome and metabolome. This example is to show how you can use Jiva studies and twin studies to explain inheritance about intermediate phenotype, in this case metabolomics. So they use the different lipoprotein fraction and various amino acid and other metabolites that they measured by metabolomics and combine that with the Jiva's results and also twin studies to predict what the variation is explained by these genes and how much is heritable and how much is environmentally contributed. So this is another example of how to combine different other platforms to metabolomics. How about the transcriptome? This is another core-affiliated work. In this, cancer cell lines from different tissues, 57 cell lines, were interrogated for both transcriptome and metabolome. And this, I'm just showing you the glycolysis genes and metabolites that they looked at. The green circles are direct connections that were already established in the HMDB database, which are directly correlate with the appropriate metabolites. And the color range, orange being 1 and black being 0, is a correlation that was seen in the cancer cells that they were studying. So this is another way of vertically integrating different omic platforms. And finally, proteomics. This is, you can see the poster outside of this study. They were studying anaplastic lymphoma kinase, which phosphorylates pyruvate kinase, therefore causing changes in the TCA cycle, glycolysis cycle, shunting to the nucleotide metabolism. The green lines are the findings from the phosphoproteomic study that they did, and the green and red dots are from the metabolomic study untargeted that they did. Here's a good way of visualizing adding strength to your biological hypothesis by integrating platforms. So I conclude by saying many things are possible with metabolomics. It's a need for careful planning and understanding what is necessary. And definitely, please insist on a very well-described standard operating procedure for collecting samples and making sure there's no variability across your experiment. There's a lot of time for questions. If you feel that you need to ask that separately to me, I'm available all day. So I'll take any questions at this time. Yes.
One of the aspects that I think is going to be very difficult to control with human studies is the duration of fasting. Mm-hmm. How can one get around that? I work in a perioperative environment, and mm-hmm. you know, we tell patients to come in, and they all have different fasting period. So if you're saying that fasting could be, the duration of fasting could be a big factor in terms of what we find, how can one get around that?
The usual recommendation is 12 hours. Human nature is to not listen to instructions, so I don't know how to get around that. But definitely instructions should be, your last meal should be at. So that's the way to get around it, but there's no way to ensure, you know, exact compliance to that.
It's particularly difficult for me because I work with children.
Yes, and, yes, I was going to say.
Yeah, adults are
and over time too. So, but definitely if they are deviating from protocol, recording that.
Okay.
But if you feel that that's causing variation in a data set, plugging that metadata into the system might help you delineate, take that variance out. So if you cannot standardize very well because of the nature of the experiment, at least record the metadata really well. So if that's the issue that's causing the trouble, we can try to remove that interference at statistical level.
Together. Does it make sense in terms of methodology by itself? If we do proteomics on cell mm-hmm. and metabolics from conditional medium or expert medium, mm-hmm. does it make sense in terms of methodology?
Yes, because the point at which you merge the two technologies at the pathway level, as long as you prove that the pathway is up or down in different methodology but related model system, you're still adding strength to your biological question.
We did analyze the data sets. by using this kind of study. Proteomics from on cells, metamorphosis on media.
Is it difficult? It's a little more complicated than the routine things, but it's doable. These are large data sets that you combine at the pathway level, yes.